Hey everyone, in this lecture we finally get to protein folding and sequence design. So we're going to be modeling proteins in various different ways, and then focusing on one or two ways to help determine the most probable sequence. So this lecture is pretty long, so it's split up into multiple parts, but there's going to be sections on a two-state model of an alpha helix for helix to coil transitions, and then the HP model for protein folding. So where do we start? Let's quickly go through the main points from the previous lecture. It's going to be short because this lecture is already long. We're then going to jump straight into protein folding, what makes them fold, and how that gives proteins their secondary structures. This means we'll be talking about alpha helices and beta sheets. From there, we're going to talk about the alpha helix to coil transitions by presenting a two-state model of the alpha helix. Then we're going to go through sequence predictions, which will require some math, in fact, quite a bit of math and a bit of statistics, then my favorite, protein-like sequence design. And once we've covered all of that, we're going to look at the energetics to see how we can model that through entropy and energy landscapes. So let's quickly recap some key points from the last lecture. Basically, what are proteins? How we can break them down into amino acids and categorize these building blocks. And then we're going to take a look at Ramachandran plots to determine the most likely bond angles. So from last lecture, we know that proteins are a class of macromolecules made up of amino acids. It's the protein sequence that's going to help determine its 2D and 3D structure, and any misfolding can cause medical issues and illnesses under a branch of illnesses called proteopathy. There are 20 amino acids, and they can be categorized into polar and nonpolar amino acids. Polar amino acids can then be subcategorized into negatively charged, positively charged, and uncharged polar amino acids. But for the most part, we're just going to say that they're either polar or nonpolar. So these amino acids, when connected together with peptide bonds, labeled here in the middle, they form polypeptide chains. You'll see here that depending on the bond angles, different R groups of the different amino acids are going to interact favorably or unfavorably with each other. So then, in the previous class, we took a look at bond angles. So for each alpha carbon, here labeled as C alpha, we found that you can rotate the left and right amide planes 360 degrees on either side, creating numerous possibilities of protein folding configurations. But which one's more likely? To answer that question, we introduced the Ramachandran plots. So darker areas would represent bond angles, so your phi and your psi values that were the most stable, and the larger the blob, the more abundant those types of bond angles would be. So now let's dive into protein folding. We're going to first talk about what forces help proteins fold and stabilize them. And then we're going to look at what types of secondary structures are formed, specifically alpha helices and beta sheets. Then we're going to take a look at some higher level structures like tertiary structured proteins. So to start off the discussion, we should talk about why proteins fold. Primarily they fold because of weak interactions. But what kind of weak interactions? Looking here in this image, we have a central protein and three zoomed in areas. So here you have electrostatic interactions or attractions from ionized side chains. So there's a negative and positive area throughout the protein that are going to interact with each other. Next, you have van der Waals attractions between close distance atoms and molecules. And finally, you have the hydrogen bond, which we've studied a lot of. A combination of all these weak interactions will create a strong bonding arrangement for every single protein, which makes them unique. Now, let's introduce something called hydrophobic forces. These are the forces that help proteins fold into compact conformations. By its name, we can probably assume that it has to do with hydrophilic and hydrophobic interactions, and that's exactly what this is. Polar amino acid side chains will want to be together, and nonpolar amino acid side chains will want to be together. With our previous analysis into polar and nonpolar interactions, water will want to be around polar molecules, and due to higher entropy, and water will want to group nonpolar molecules to minimize their individual interaction with them. So this means that nonpolar side chains will want to be essentially buried inside a protein conformation and then create something called a hydrophobic core that makes the entire protein highly packed. So essentially think about the whole protein trying to fold in on itself so that it gets this strong hydrophobic core of nonpolar amino acids. So if you end up with a protein like this, with a hydrophobic core, all of its hydrophilic polar side chains will be oriented on the outside of a protein, and while all of its hydrophobic nonpolar side chains on the inside. So then, once your protein's folded into its desired shape, or its most probable shape, what keeps it together? Specifically, it's the hydrogen bonds. They occur between the polypeptide backbone and or its side chains to stabilize a protein's folded conformation. So you have hydrogen bonds occurring from backbone to backbone, from backbone to side chain, and from side chain to side chain. So this helps to stabilize a protein's folded conformation. And now we want to look at how proteins can be guaranteed to fold properly, because there can be so many different conformations that they have to guarantee their perfection somehow. So let's now introduce a type of protein that we've talked about before. They are called chaperone proteins, and these assist in protein folding. These chaperones make sure an isolated polypeptide is folded correctly and doesn't aggregate into non-functional structures. Looking at the diagram here, I have a whole bunch of, semi or partially folded proteins, and if it's not folded properly, there's a high chance that these partially folded proteins can clump together and create something that doesn't work well. So each one of these individual partially folded proteins can go into a chaperone protein. And each chaperone protein is going to provide a clean environment to allow the protein to fold correctly all by itself. And then after it's folded, it gets released. So you get this perfectly folded protein that's released from the chaperone protein after the folding process is complete. So hopefully this makes sense. But the other thing I wanted to talk about were heat shock proteins. Something unique about chaperone proteins is that a lot of them are heat shock proteins. So heat shock proteins are created because of stress. So you've got high temperature, UV radiation, etc. Because of stress, there's a higher chance that proteins can get denatured or misfolded. So heat shock proteins are connected to chaperone proteins because the chaperone proteins are there to guide protein folding and fix protein folding. So as this level of stress increases, the need for our chaperone proteins increase. So the majority of chaperone proteins are also heat shock proteins that show up when there's levels of high stress. Now let's move on to what happens after a protein folds. It goes into its secondary structures. So you've probably learned about alpha helices and beta sheets. These basically result from hydrogen bonds that form between the amino and carboxyl groups in the polypeptide backbone. On the left, you can see an alpha helix, while on the right, you see a beta sheet. So we're going to take a look at each of these individually for the next few slides. Looking at the alpha helix, we have hydrogen bonds between the NH and CO groups of the polypeptide backbone. So they're represented by these red dashed lines here. And we can also represent this as a helical structure, with each turn being 0.54 nanometers, and usually it includes 4 amino acids, or 3.6 residues to be exact. So here's just another look at the hydrogen bonding arrangement. We can see that they occur in a stepwise fashion. So the first monomer is going to bind to that monomer plus 1, 2, 3, 4. So monomer 1 will bond to monomer 5. And we can see that they occur in such a fashion where these types of hydrogen bonds is going to allow for a patterned folding of an alpha helical structure. Now, the next thing to mention is that a helix can be left-handed or right-handed. So how do we know which one's more common? Maybe now it's time to take a look at the most probable bond angles, and we can do that through a Ramachandran plot. So this Ramachandran plot should probably be very familiar to you. It's the same one that we've been using the whole time. And basically we now can put some examples to the test. This plot tells us what kind of bond angles are the most common. So if the bond angles are phi equals 60 and psi equals 60, seems like a left-handed helix can occur, but the light color of this area tells us that it's not very common. Now we can look at the -60 and -60 bond angle pairs, and there's a much greater area that's darker, telling us that this is a more stable and abundant conformation. So when we're comparing just the left-handed and right-handed alpha helices, the right-handed alpha helices, we can say they're going to be much more common than the left-handed ones. There's also this remaining dark blob that's up here in the top left corner. What could this mean? Basically, there has to be another conformation we haven't looked at yet, and we're going to take a look at this soon. Now let's first talk about what alpha helices are useful for and what they can do. First, an alpha helix can cross a lipid bilayer, so that's very useful. And why is that? Because it has hydrophobic side chains, which can insert into the hydrophobic core of the lipid bilayer. So they're going to like being together. Remember, there's a difference between proteins folding so that they can have their own hydrophobic core and the conformation of an alpha helix. So these types of alpha helices with hydrophobic side chains will easily insert into a lipid bilayer. Next, the helices can also intertwine with each other to create something called a coiled coil. And this efficiently minimizes the exposure of hydrophobic residues. So the next secondary structure we want to talk about is the beta sheet. Once again, it's formed due to the hydrogen bond, but arranged differently between the amino and carboxyl groups of the polypeptide backbone. So instead of forming a helical structure, it forms a sheet. So there's different layers of beta strands that are created. So your beta strands will now make up the last blob that we saw on that Ramachandran plot. This tells us that beta strands are going to be much more favorable and stable conformations than your alpha helices, and whether it's left or right-handed. But just like alpha helices have left and right-handed versions, beta sheets also have different directions as well. This plot only shows us the beta strands, not how the strands interact with each other to create the beta sheets. So when we take a look at the beta strands and connect them together, we notice that the sheet can be anti-parallel like this. So we have sheets going here, and then in the opposite direction. Or it can be parallel like this. And if it's parallel, we can note that the hydrogen bonds are going to occur between alternative amino acid side chains in order for them to face the same direction. So finally, after you have had alpha helices and beta sheets, they can combine and form protein domains. So these domains can come together to form larger protein aggregates or protein molecules. And the final form is called a tertiary structure and usually has different functional domains. So in different colors here, there's one functional domain and another functional domain that has different functions and uses throughout the body, for example. So in terms of these functional domains, they can be a combination of different alpha helices, different beta sheets, or both. So now we can move on to modeling all of these. There are going to be several models that we present, some visual, some mathematical, so the next few sections will be quite long, and they're going to be broken up into different sections as well, so it's not too overwhelming for one view. So first off, we're going to start with the two-state model of an alpha helix. We want to explain something called a coil to helix transition, and then use that knowledge to present a model that we can apply to predict the hydrogen bonding order of an alpha helix. At the end of this, we're going to present a sample problem of hydrogen bonding sequences. So from before, we note that a right-handed coil was more favorable than the left-handed coil. Kind of drawn it here. And we also learned that it was stabilized by hydrogen bonds. So why don't we just take a look at the individual subunits and assume that they're all in unity. So basically, we have a homogeneous strand of polymer beads that we're connecting together. But the one thing we want to change is whether they're in a helix or a coil state. So what does this mean? When looking at a homogeneous polypeptide chain, we note that there can only be two forms for each monomer. So each monomer can either be in a coil state, where the bond angles are free to rotate and therefore have larger entropy, or the monomer is in a helical state, which means that the bond angles are essentially frozen because they're stuck in a helix structure. This is going to force it into a lower energy state. So when a monomer changes from a coil to helix or helix to coil state, we call this a coil-helix transition. And of course, this then means that it's a two-state model of an alpha helix. And this is going to allow us to make various predictions about which conformations are possible or not. So then how do we do that? Let's first allow each monomer, so we represent each monomer in blue, to be either in a helix or coil state. And we designate these as H or C. You'll see in the diagram here that there is a polypeptide chain that I've drawn, and we can represent each amino acid, so 1, 2, 3, 4, 5, as a blue circle labeled in the order of the chain. There's also a green dotted line to represent a hydrogen bond between monomer 1 and monomer 5. And we can represent this in sequence form as well, like this, or like this, with an overall conformation of 1H. So note that there is going to be one hydrogen bond and that it's connected between the first monomer and the fifth monomer. And in order to write this down as a sequence, we say it's H, CCC repeated for however many monomers you have. Remember, there's only one hydrogen bond, so it's the first monomer that initiates that hydrogen bond, and therefore it's only the first monomer that we can say is in a helical state, because this one is bound to the top monomer 5 in a helical structure, but number 5 does not have another hydrogen bond above it, so it's still in a partially coiled state. So with our first model, we made the first monomer hydrogen bond with the fifth monomer. So what happens to these three over here? We call them intermediate monomers, and they're essentially in a coil state that's kind of stuck in a helical state. So if we think about whether they're happy or not, essentially they are still in a coil state and should be free to rotate. But in a way, they're not comfortable. They're already slightly ordered because of the existing hydrogen bond, forcing them to bend like that. So they want to rotate, but they can't, which means these are energetically unfavorable. So we now ask the question, what is the next favorable conformation? If we have our sixth monomer, so if we add one more monomer up here, what is it going to do? If we think about this together, the sixth monomer will most likely hydrogen bond with monomer number 2, the second one going along the chain. And this is going to be favorable because this conformation doesn't require the ordering of one extra monomer. Number 2 is already in a prime position for a hydrogen bond. So here I've drawn our 6th monomer and have connected it to monomer number 2 using a hydrogen bond. And we can now represent this protein sequence such that we have 2 monomers that are hydrogen bonded, and you have HH and then a whole bunch of C's afterwards. Just to follow up with that, I want to reiterate the fact that forming the second hydrogen bond does not require the ordering of an extra monomer. Therefore, it makes sense that it's just the first hydrogen bond here that's the hardest to form, and then the others just fall into place because they're already energetically unfavorable as they are. And because you have more energetically favorable conformations and hydrogen bonding occurring throughout the whole alpha helical structure, we call this the cascade effect, where each additional hydrogen bond will take one extra monomer until none are left. So here in this diagram, we've connected all of the monomers, 1, 2, 3, 4, with all of the monomers above, so monomers 5 to 8. And there are a total of 4 hydrogen bonds now, so we can say HH, HH, and then all the rest are C's. Here's another representation of the sequence with hydrogen bonds. So it's another way to represent it. You just have a stepwise fashion of hydrogen bonding, where the first one is matched with the fifth one, so there are 3 intermediate monomers between each hydrogen bond. So now would be a good time to go through a couple examples of what would or would not be a favorable sequence. In this example, it's asking us if the given sequence is highly possible or impossible, and to explain why. So looking at this, we have hydrogen bonds between 1 and 5, and 4 and 8. So it's skipped 2 and 3 entirely. So why don't we represent this on the 1D sequence here as a connected hydrogen bond between 1, 5 and 4, 8. So we have something like this. We're going to take the first monomer in the 1D sequence that is hydrogen bonded as H. Then we get CC, another hydrogen bond starts at monomer 4, so that's in the helix state, and the rest is C. So is this favorable? The answer is no. Looking at the sequence, we have 4 monomers in red that are not hydrogen bonded and want to freely rotate but can't. So it's highly energetically unfavorable, and therefore the sequence is highly improbable. Let's take a look at a second example. We now have 1, 2, skip 3, and 4 that are hydrogen bonded above. And we can fill in the information like we did before. So we're left with 2 red circles, which represent intermediate monomers that are energetically unfavorable. So in this case, can we ask whether it's, or can we determine whether this is more energetically unfavorable than the previous conformation? So looking at this, we already know it's an impossible sequence. But these particular monomers are even more uncomfortable. They're in the coil state, but because there's hydrogen bonds all around it, it's forced to be in a very stable position already. So that means it's highly energetically unfavorable, but because it wants to rotate, but essentially it's locked into place already. So what if we take a look at a full helix? What would the sequence be? From our examples before, we had 4 monomers in one ring. Going from bottom to top, we had the bottom 4 monomers being hydrogen bonded to the ones on the top. So this would mean every monomer is hydrogen bonded for a full helix, except for the top end of the helix. So we would basically have all of these monomers initiating hydrogen bonds with their upper monomer, except for the top one because there's nothing at the top. So this would mean every monomer is hydrogen bonded for a full helix except for the top end, and you end up with an overall conformation with 4 C's at the very end. So then, looking back at everything that we've covered, my question is, why did we have monomer 1 bind with monomer 5? And it's because every turn of the alpha helix has 3.6 residues. So to make things simpler, using an I plus J or I plus 4 or J plus 4 spacing allows us to add 3 more atoms to the hydrogen bonded loop compared to a tighter helix, and provides a better model for us to study from. So if you remember this image from before, we now have a hydrogen bonding pair here. We can call this monomer in the Jth position, and then the bonding pair will be in the J plus 4th position. And that means there are 4 subunits per term to accommodate for the 3.6 residues per turn. So why do we even care about this? We care about this because we want to be able to figure out how many helix states there are compared to coil states in a solution. So this is just a diagram of some helix states and coil states. And the simplest model of this was to just create a one-dimensional sequence of H and C subunits. But the goal is to be able to compute how many of these proteins are in helical states and how many are in their coil states. So in that case, we have to compute the partition function, and that's going to be based on the combinatorics for counting the numbers of arrangements of H and C units in the chain. So we're going to talk about cooperativity in a bit, but let's first look at this. When we talk about a cooperative trend, it usually means a sigmoidal curve, something like this. And there's going to be different curves for different lengths of polymers, which we call N-mers. We can kind of see that the longer the polypeptide, so the more dots, the more monomers there are, the longer the polypeptide chain, the steeper and more pronounced the sigmoidal curve. And what does this mean? What we can get from the graph is the following. The larger the polymer chain, the more sensitive it is to temperature changes. The theta here represents fractional helicity, and we haven't covered that yet, so we won't worry about it. But for now, at least we know that temperature is a factor that affects the cooperativity or the helix to coil transition for proteins. So now it's time to put everything we've learned together through some mathematical models. Essentially, these are models that govern phase transitions. Small changes in temperature or a solvent can cause a polymer molecule to undergo a helix to coil transition, such as causing 2 strands of DNA to zip together or cause a protein or RNA molecule to fold. So we're going to introduce a term called subunit cooperativity, which can be positive or negative. Basically, what this means is whether it's easier or more difficult for something to happen involving multiple units. For example, positive cooperativity amongst adjacent DNA nucleotides is going to allow for easier unwinding of a whole group of adjacent nucleotides. So that would be for DNA unzipping. If you have positive cooperativity amongst adjacent amino acids for hydrogen bonding, that's going to allow for an easier helix to coil transition or vice versa. So why don't we start with some of the models that we can use? Let's start with the Lifson-Roig model, which you've probably seen before. It's the one we just covered in the previous section. We're basically going to assign each residue in a polypeptide a helical or coil residue state. So you either have H or C. So this is called the Lifson-Roig model because it is a statistical weight model. Before, we simply just used H's and C's. The Lifson-Roig model applies a model to allow us to calculate the probability of certain conformations. So what we're going to do with the statistical model is we're basically going to assign each residue in a polypeptide a helical or coil state. So it's a 1D sequence model, and that means we assume the total number of possible states in an N-mer with N residues will have 2 to the N molecular states that are possible. And now we want to ask what happens if we assign each of these individual residues a statistical weight. This means that the product of these weights will give rise to an overall statistical weight for a specific configuration of a polymer with N residues. So speaking of statistical weights, we'll have to start assigning them using the LR model. So we want to define 3 different residue weights. We have U being the residue weight assigned to a coil state. This is straightforward because there's obviously no hydrogen bonding, so it's just U or with hydrogen bonding, which we represent with V. When we look at helix states, it does get a bit more complicated because the helix state monomer is either alone, so it has no adjacent monomers that are hydrogen bonded, or it does have neighbors that participate in hydrogen bonding as well. And that's going to change whether that's going to change its weight and its energies. So if we think about them a bit more, these statistical weights are based on local interactions. We have U being a coil state. If it's a coil state, then it's a coil state. There's not going to be any neighboring monomers that are hydrogen bonded. If you're looking at helix states, either itself is hydrogen bonded with a coil neighbor, or so we represent that with V, or we use W to represent the weight assigned to a helix state with no coil neighbors. Oops, I probably mixed the two up. V is the one with coiled neighbors, and W is the one with no coil neighbors, so everything is hydrogen bonded. So if we want to calculate the weighted average of that property over all states, we have to use a summation. So that summation term is given here, where we're going to say the weighted average A is the sum of every single state's probability times its weighted value. So we have this equation. Now where do we go from there? We end up with something called the partition function, Z, which describes the statistical properties of a system in thermodynamic equilibrium. So first of all, it's a dimensionless pure number, and for our purposes, we want to focus on the canonical partition function. What this means is that the system is allowed to exchange heat with the environment, but the system is at a fixed temperature and a fixed volume. So the definition of canonical partition function is given by this summation over here, and you'll notice some terms that are quite familiar. We know that S is equal to KB ln W, and if we solve for W, we get this term, S over E to the power of the Boltzmann's constant. And through an exercise we did previously in the entropy chapter, we were able to convert this into this expression. So we added a T on the top and bottom, and we're able to redefine the S at the top to be negative F divided by KBT. So once we rearrange this, we know W is equal to or approximately equal to this term. When we can plug that into here and get an equation for our partition function. So this is great because what we can do is assume that the weight of every state is going to be equal to its multiplicity W. So essentially this is just the summation of multiplicities. So remember the 3 states we had before? We had U, V, and W. So basically coil, helix around coil, or helix without coil neighbors. What if we simplify this to 2 possibilities instead? So this is the LR model, so we want WC for the coil state and WH for the helix state. Just want to separate them into two. But we can't forget that the helix state can have a coil neighbor or a non-coil neighbor. So we now have to determine what factors play a role into each state's multiplicity. When we look at the coil state, it seems like freedom of rotation is going to play a huge role. So we'd probably take a look at bond angles. And those bond angle conformations mean that your weight of your coil state, that's going to be the primary decider of how much statistical weight you have. Next, when we want to look at the helix state, they're governed by hydrogen bonds because either they have hydrogen bonding neighbors or they don't. And there's also a hydrogen bond free energy, epsilon, which we'll define here, that we can talk about later on in this chapter. The one caveat of all this is that we have to divide all these states by the weight of C. And it would be good to understand why, because it just makes our lives a lot easier. So if we divide all the states by the weight of C, what this is going to do is give us a reference point for further analysis and comparison. So that means if I divide the weight of C by the weight of C, I just get 1. So we can assume WC is equal to 1. Makes things a lot easier and straightforward. When we take a look at the weight of a helix state, we have to consider two options, just like before. So instead of looking at the V and the W, we now have the weight of H, but we just multiply it by a factor to see what its neighboring monomers are like. So for every H that comes after an H, the weight is S. But for every H that comes after a C, we use sigma S. Why sigma? Well, if we think about it, we had looked at the helix coil transitions before. The chances of getting an H after an H or a C are going to be different. So we have to introduce this variable to be able to differentiate between the two. But are the chances of getting an H after a C greater or less than the chances of getting an H after an H? We've shown previously that it's easier to get a helix state immediately after an existing helix state. So that means the chances of H after C will be much, much lower because that's considered the first hydrogen bond, which is the hardest one to make. So this means the value of sigma, because it's defined as a parameter for every H occurring after a C, this statistical parameter must be less than 1 so that the statistical weight of H after C is less than H after H. So that forces this statistical weight to have a value less than this one. Overall, this makes sense because the first hydrogen bond will always be the least favorable and the least possible. And in fact, when we take a look, the value of sigma is really, really low. So you get a value of sigma between 10 to the negative 4 and 10 to the negative 2. Let's now take a look at a dimer as an example. So that means just two amino acids. We can obtain a list of all possible sequences of a dimer by combining them. We can have CC, CH, HC, and HH. So these are all the possible combinations. The weights can also be defined. You have 1 times 1, 1 times S times 1, and S times S. So these two are going to be the exact same weight because it's the exact same makeup. So we can also make our lives a lot easier by using a binomial expansion to quickly get all the possible sequences. So if I have C plus H to the power of 2, because I have a dimer, I have C plus H times C plus H number of sequences, and I can actually get all the terms out right away. So the next thing to note is that the probability of your entire sequence is going to be proportional to the weight of your entire sequence. So these are just some tricks or tools that you can use in case you ever have to figure out all the possible sequences for, let's say, a trimer or an N-mer. It'll get a lot worse the longer your chain gets. Now let's take a look at the probability of each sequence. We can simply divide the multiplicity of that sequence by the total multiplicity to get the probability. And we can get the following expressions for each one of our probabilities. So this is the probability of getting 2 C's, or as we call it, 0 H's. This is the probability of 1 H, and this is the probability of 2 H's. So these are different expressions that you can get using this equation, and it would be good practice to try and work your way through it so that you can get to the answer. The only thing here is that the denominator seems to be very cumbersome. There's a lot of terms, so maybe we can use a different representation or simplify it a little bit. So let's revisit the partition function z and introduce it here. If you actually calculate z2, or the partition function for 2 states, you get 2 monomers, you get 1 plus s to the power of 2. So we can simplify the denominator in any of these 2 ways. You can write it as z2 or represent it as 1 plus s to the power of 2. And then we now want to define fractional helicity, which we introduced before for the graph, and we weren't able to explain it before, but we can now. That's the ratio of helix states to the total number of residues or monomers in the sequence. Let's go through an example to try and find the fractional helicity of a dimer. So in order to find the fractional helicity of a dimer, let's first calculate the number of H's that can occur. We now know that there are 0 H's in the CC state, but 1 H in the CH or HC states, and 2 H's in the HH state. So all we really have to do is just multiply these values with their probabilities. There are 0 H's in the CC state, so basically this whole term goes to 0. There's 1 H multiplied by the probability of these sequences, plus 2 H's times the probability of this sequence. So if you do the calculations, you end up with 2s over s plus 1. However, we know that we have a total of 2 monomers, so we divide everything by 2, and we get s over s plus 1 for the fractional helicity of our dimer. We're not quite done yet. Let's take a look at an analysis for an n-mer. So to make things easier, probably good to use the binomial expansion to find all of your possible sequences. Then let's say you want to find the multiplicity of one single helical state out of n states. So you would then have to use this equation. So you have your multiplicity times your s. And you can represent it like this and find all the possible weights for every single sequence that's possible. As you can see, very time consuming. After you figure that out, you can find the partition function as the next step, which ends up just being 1 plus s to the power of n for expansion. You can then get a general term for the probability of an n number of helical states. And then you can find the average number of H's using a summation. But we can represent one of these terms as a partial derivative. So keep in mind, if we rewrite all of this, ns to the power of n is equal to s times the partial derivative with respect to s of s to the power of n. So if I rewrite this as this partial derivative, we end up with this expression. So continuing on with that expression, we move on to the next slide, and we end up with this term. You can move some of the constants out, and if we take the partial derivative with respect to x of the entire partition function, we can rewrite the partition function as ns over s plus 1. So if we want to find the fractional helicity, all it is the number of H's divided by the number of monomers. So that's this term divided by n, which ends up being s over s plus 1. And this is great because the value is the exact same as a dimer, and we can assume that the fractional helicity is going to be proportional to s over s plus 1 for any polymer that we have. So let's try and interpret what we've derived. The fractional helicity for sigma equals 1 is given as s over s plus 1. The parameter s is called the relative weight for h, where we can say it's the weight of h divided by the weight of c, like we've defined previously. This means that we have the hydrogen bond free energy up at the top and the multiplicity for c down at the bottom. So thinking back to the graph we had before, can we now relate this to temperature? And the answer is yes. Because we have this kBT term in the exponent, there is a direct connection between fractional helicity and temperature. But before we can even understand that graph, we should define a few more things and figure out where that bottom term, or not bottom term, the x-axis, which had T minus Tm, we want to see where that comes from. So let's ask ourselves whether a given polypeptide will assume a random coil or alpha helical structure. And it really depends on the competition between conformational energy and hydrogen bond formation. This means we have to take a look at the polypeptide's composition, thermal environment, and chemical environment in order to figure this out. So we usually test this using polarized light to get optical rotation values, where we can see a total conversion of a sample from one form to another upon a temperature change of a few degrees. So now we can finally take a look at our graph, or a similar graph that we looked at previously. You can kind of see that at low temperatures, all the samples displayed an optical rotation similar to that of isolated monomers, or basically random coils. At high temperatures, though, the rotation changed. So the top dots are polymer chains with 1500 monomers, middle dots have 46 monomers, and the bottom dots have 26 monomers. So we notice that the larger the polymer chain, the sharper the transition from random coil to alpha helix. So that tells me that larger polymers are more sensitive to temperature changes. And now we want to ask how sigma factors into all this. Remember, sigma was the probability of getting an H after a C. So if we have a full alpha helix over here, the chances of getting an H after a C are pretty much 0. So the transition becomes sharper as well. So remember, sharper curves are going to come from a larger temperature and a smaller sigma, because that means it's going to very quickly switch to a helical structure. So that was Zim and Bragg's data fitting model, and we presented a graph before. It wasn't optical rotation versus temperature, but rather fractional helicity versus T minus Tc. So we should probably see where it comes from. Zim and Bragg were researchers that discovered an expression for the parameter s, and that s was approximately equal to e to the power of 0.00614 T minus Tc. And they experimented with this mixture in order to get those values. So when you have T minus Tc, the logarithm, oh, not the logarithm. When you have T minus Tc, that's the difference between the temperature and the critical temperature, which is the magnitude of the midpoint temperature. So we're going to go through that in one of the assignment questions, what the midpoint temperature is and how you can actually calculate these. But for now, we're able to rewrite the equation for fractional helicity. So you had s over s plus 1. We can represent it and tie it back to temperature T. And this, if you plot it, gives a sigmoidal curve. For different values of n, you get different steepnesses of each curve. So looking at Zim and Bragg's experiments, we noticed that we had to increase the temperature relative to some midpoint in order to increase the number of helical structures. But why? It's because each act of hydrogen bonding releases solvent molecules that are previously bound to the chain. We know that the change in Helmholtz free energy is given by E or U minus Ts. If we assume that the change in bond energy is greater than 0, so over here, it means that at a higher temperature, the free energy will become more and more negative. So as free energy decreases, hydrogen bonding is going to become more and more spontaneous. So that's why you have that sharp increase in your curve. If anyone noticed, we previously said that sigma should be really small, but we use sigma equals 1 for our calculations for simplicity. Using that simplification, what would be the total weight of a trimer? So let's try to solve this together. Won't go through all the steps because that would probably be part of an assignment question. But to start off, to find the probability of each conformation, we need the partition function z in our denominator. You can do the calculations for the partition function and end up with this solution in green over here. But there's a much better way to calculate Zn, especially as n gets larger, and that method is called matrix formalism. So keep in mind this isn't a tool you have to use, but it's there if you choose to do so because it does help quite a bit. So we should probably go through what matrix formalism is. The Zim-Bragg model basically takes the cooperativity of each segment into consideration when calculating fractional helicity. You can write the full partition function for a chain of n residues using these matrices. So you have CH in the first one to represent the statistical weight vector. So that's how much each of these states is worth or weighted statistically. Your 2x2 matrix is going to represent the monomer's conditional statistical weights. So that would be your 4 possible combinations of C and H. You then multiply by a column vector of 1s to sum over the statistical weights of all of your sequences. And you're going to get the same answer here in a lot less work. So now let's take a look at hydrogen bonds in water. Any molecule that can form hydrogen bonds can alternatively compete to form hydrogen bonds with water. And what does this mean? It means that the hydrogen bonds formed between two molecules dissolved in water are relatively weak because there's always competition for hydrogen bonding. So that's something that we looked at in our assignments. To wrap up this section, I want to present something that makes a helix not form into its usual shape, so an exception to the rules. Let's introduce proline, represented as P, and it's known as the breaker of helices. It actually helps to stabilize a hydrogen bond between the first and third monomers versus the first and fifth monomers. This causes the helical structure to break and could be a cause for misfolding. So we've gone through coil helix transitions, which were pretty 1D or 2D. We're now going to go through a completely different model for protein folding in general, something called protein-like sequence design, which considers the 3D structure of proteins. We'll wrap up with a discussion of some energies of protein folding, so that would be energy landscapes, which is very important to understand. First, proteins can assemble into different sorts of shapes, like dimers, helices, and rings. When these complex proteins connect through binding sites, different conformations are possible depending on the shapes of these binding sites. So, looking at this example here, we present 3 different types of binding sites. There's this one with just a little concaved area here, and when 2 of them stick together, it forms a dimer. And they're pretty limited in the types of structures they can form because of the shapes they're in. You can also have these this type of protein with 2 binding sites. So in this case, it makes sense that it could be a chain of polymers, in this example a helix, or not a chain of polymers, a chain of monomers, creating a helical structure, or essentially just a long strand of subunits. If you have binding sites that are kind of at an angle to each other, you can create ring or circular structures as well. So the type of binding site that you have is going to really help determine what kind of conformation is possible. An example would be fibrous proteins such as collagen and elastin, which are elongated fiber shapes. Collagen is a triple helix formed by 3 protein chains, while elastin has molecules that are cross-linked together and can be stretched here or relaxed in this form. So just keep in mind, the collagen triple helix is just a tiny short section of the collagen fibril, whereas the elastin is going to help with elasticity. Now remember we had mentioned proteins are stabilized by hydrogen bonds. That was in reference to individual protein chains. For multiple protein chains and complex conformations, the presence of disulfide bonds here can help stabilize these. So disulfide bonds are the most common covalent cross-links and help to reinforce a protein's shape, like this, to maintain or retain its activity for a lot longer period than if it weren't there. So why do we even bother with 3D structures? Because it allows proteins to interact with other molecules. When protein folding occurs, there's usually cavities on the protein surface that we call binding sites. So we briefly talked about this before, but now we're going into a little bit more detail. These binding sites are going to contain a set of amino acids, side chains, that can bond only with certain ligands. So using this as an example, you have a folded protein. Essentially, you could have a hydrophobic core or not. These are presented to the surface. It's a concave area with a bunch of amino acid side chains sticking out. If we take a look at the next diagram, we call any binding in these concave areas or binding sites a key lock mechanism of binding. And this has everything to do with biological recognition. Just like a specific key can fit into a lock, a specific ligand can fit into the specific binding site, so that a specific combination of shape and weak interactions will be present for the perfect match. So here, cyclic AMP is bound to a folded protein. It's only this type of ligand that can actually bind because it forms perfect connections and hydrogen bonds with all of the amino acid side chains that are presented at the surface of the binding site. Another great example would be antibodies and antigens. An antibody is Y-shaped and has 2 identical binding sites for its antigen, one on each arm of the Y shape. An antibody is going to be composed of 4 polypeptide chains, 2 heavy and 2 light, which are held together by disulfide bonds. So why don't we take a closer look? We have the Y shape for our antibody, and we note that there are 2 long chains, so the 2 long chains are attached at the center, and they're called the heavy chains because they're bigger and longer. Then you have these smaller arms called the light chains. Within the light chain, so zooming into one of these light chains, you have a variable domain up here and a constant domain down here. The image here on the right shows that there is a constant domain which remains the same, and at the tip of the light chain you have what's called the variable domain that can change, and that's important because it allows the antibody to have different antigen binding sites for molecular recognition. Keep in mind also that the heavy chain also has a variable domain up at the top. So if you have two different types of variable domains at the tips of these two chains, you can have many different combinations for different antigen binding sites. So then let's compare the entropy, energy, and stability of a folded versus denatured protein. When the entropy is large, we're assuming that the protein is denatured because there's a large number of configurations possible, and it's pretty much like spaghetti. It's not ordered in any way whatsoever. A folded protein will have much lower entropy because it's highly organized and therefore many more favorable contacts. This lower entropy means a higher level of organization and therefore a more stable conformation. So let's now move on to protein folding models. That's some of my favorite stuff. We know from our previous discussions that proteins have to be in the right shape or else they can't function. So why don't we consider two types of proteins, folded and denatured? So this is technically coil-like, and this has been collapsed into a compact protein. In the denatured option, using our two-state model, the multiplicity of a coil conformation is very high, right? You can have many different conformations of this strand. In the folded option here, the multiplicity should be much lower. In fact, if you can only have a protein fold into the correct shape in order to have some sort of function, it's almost required that the multiplicity be 1, so that there's only one preferred structure. So we call this a multiplicity of 1 because there's only one possible shape once the protein is folded that will impart a specific function for whatever it's trying to do. So this is going to be very important when we start to talk about degeneracies, which have to do with the introduction of energy levels as well. Now, in order to create our protein folding model, we need something to focus on. And remember in our earlier discussions that hydrophobic forces were a main driving force between protein folding. So we're now just going to solely focus on this versus hydrogen bonds, because hydrophobic or nonpolar amino acids will want to be buried inside a hydrophobic protein core. So they'll always want to go inside. So to recap, we know that a proper folded protein has a multiplicity of 1 because there's only one good preferred shape, and that hydrophobic forces are at play to stabilize protein conformations. So how can we actually model a protein chain in 3D? How about we use random walks along a lattice? If we can use compact Hamiltonian random walks, that's a good start because it simplifies everything quite a bit. We can model protein conformations as compact random walks that make contact with amino acids at the surface of the protein only, and we can assume that every amino acid will have to take up all the spots on the lattice, so we can't leave any space empty. So what can this guarantee? It guarantees that all the solvent sites are pushed to the surface so that we only care about the hydrophobic interactions at the outermost faces of the lattice structure. And before we even go into modeling these proteins and polymer chains, the assumption is that when you have a compact structure and the protein is folded, there is going to be a hydrophobic core, or basically a bunch of favorable hydrophobic contacts at the center of your lattice structure. And then the more unfavorable contacts on the outside or surface of your structure, the less likely that particular conformation will occur. So this is just a diagram, but when we look at the homopolymer case, where all the subunits are the same, we're looking at all the different ways that the monomer subunits can be arranged. So this is a case study of a 6-bead polymer chain. So these are all the different conformations you can have with six monomers. there are tallies of energy levels on the left, and then there's a tally of the number of contacts on the right. These contacts are considered preferable contacts. So if I have a contact here and here, it means that we can assume that all of these beads are hydrophobic, and hydrophobic beads or monomers are going to want to stick together. So there's 2 preferred contacts for all these, and we call this a ground state of energy. There's 1 favorable interaction here. We call this an additional energy level. And we have 0 contacts here, which we say has 2, is our second tiered energy level. So now let's look at combining everything we've learned into a better model. What we just looked at was called the homopolymer case. It means that the monomer subunits are the same. And for proteins, we want to look at the number of hydrophobic contacts. So I kind of touched on that before, but it becomes very important because remember with the previous one, there were 4 spatial rearrangements of the same protein. So these ones, 4 spatial rearrangements to achieve the same energy level. So that's technically not good because there's no unique ground state. They're all at the same energy level and there's nothing unique about it. What we're essentially looking for then is a degeneracy or multiplicity of 1. So if we can find that, then we can say that this protein sequence is protein-like and a good candidate for preferred protein structures. And then we can make predictions off of that for some preferred conformations. So instead of looking at the 2-state HC models, we now want to switch to HP models, which stands for H, hydrophobic, and P for hydrophilic. So to start with the HP model, let's first state what we want to model everything as. Remember back when we said amino acids could be polar or nonpolar? That will come into handy now because we're just going to use red for hydrophobic and white for polar. And that's what we call the HP model. Looking at the HP model, it divides the 20 amino acids into 2 classes, as we've mentioned before. Hydrophobic for all of these, and polar for all of these. And this model is useful because, number 1, it helps reduce the complexity of the protein sequence. Number 2, if you want to analyze a polymer of 100 amino acids, you can easily reduce the number of possible sequences from 20 to 100 to 2 to 100. Then if you apply a computational model to study this, it'll make the program run much faster and much more conveniently. So let's start with the 6-mer HP model, commonly referred to as the toy model. So it has 6 monomers, like this, on a 2x3 lattice. And remember, the requirements for our models is that the amino acids, or the monomers, have to take up every single space in different conformations. But we're using a compact random walk model, so we need to be able to only look at different overall shapes or structures that can be formed. So just by doing the math, we know that if we have 6 monomers with 2 states of possibilities, there are 2 to the 6 or 64 sequences that are possible in total. That's a lot of sequences to analyze, and we don't ever want to do that. So we want to look at compact structures. And if we take a look at a 2x3 lattice and require all the amino acids be linked together with no overlap, and that all the amino acids have to take up all 6 spaces in the 2x3 lattice structure, then you only end up with 3 general shapes. You have your upside down U, or pie shape, S shape, and G shape. And you may have the first one on the left flipped upside down, or like a U, or like the pie, but it's still the same shape. So we call a compact structure the same general shape to look at. So in this case, for a 2x3 lattice structure, there's only 3 possible shapes for our protein. Looking at the 6-mer toy model, the hydrophobic energy of this HP model really depends on its sequence. So what we're going to do is assign an energy cost, epsilon, for every contact. So if I'm taking a look up here, my hydrophobic molecule in red is not going to want to interact with a nonpolar molecule, and vice versa, or interact with a hydrophilic molecule here. So what this means is we just tack on all of the unfavorable interactions that could occur and assign and add them all up and give them an unfavorable contact or an energy cost. So here I have 1, 2, 3, 4, 5, 6, 7 unfavorable interactions because a hydrophobic molecule is not going to want to interact with the water or solvent around it. So that means all of the exposed hydrophobic molecules will experience at least one unfavorable contact. So the more unfavorable contacts you have, the least likely that structure is going to occur. So we're going to take a look at some examples now. Let's first start with the top sequence. Remember, red was hydrophobic or nonpolar, and white was polar or hydrophilic. So this means starting at the top and starting at the bottom left, we have H, P, HP, HP, and that's the model we have. The sequence is HP, HP, HP. There are 3 separate compact shapes that we can form with the sequence, and they're just drawn here. So the next step is to now count how many hydrophobic contacts there are so we can assign an energy cost to each of them. One hydrophobic contact is not preferred because the assumption, or any hydrophobic contact is not preferred because the assumption is that the protein is in water. Like I said before, you have a polar environment, and the more contacts you have, the more unfavorable the configuration. So looking at the U shape, we have 7 hydrophobic contacts. Counted those before. Let's count these ones. 1, 2, 3, 4, 5, 6, 7. Seems like we have 7 hydrophobic contacts for all 3 of these structures. So is there any unique ground state? All of them kind of look the same, different structures, same energy cost. There's basically no one structure that stands out. So is this a protein-like sequence? The answer is no, because there are so many hydrophobic contacts that, number 1, it's already unfavorable, and number 2, they're all at the same energy levels, so the protein probably will never be able to decide which structure is the best. Now looking at the bottom sequence, we now have P, H, PP, HP. So that gives us this sequence. And we now count to see how many hydrophobic contacts there are. This one only has 2. We can't count the contact in between the 2 of these because this is a preferred contact. We have 2 unfavorable contacts here and 2 more unfavorable contacts here. So the 2 of these shapes, S and G, have 4 unfavorable hydrophobic contacts, or you have 4 energy penalties. But this U shape, or PHYS shape, only has 2, and it's unique. So that means our U shape has the least number of hydrophobic contacts. The multiplicity of this shape occurring is 1, and the degeneracy is 1 because it is at a specific energy level that's the lowest energy cost to the whole system. And we can now conclude that this qualifies as a protein-like sequence because there is one unique lowest energy conformation that is possible. So I hope this makes sense. if you're struggling with this at all, try to take another look at the two sequences presented. Maybe try to come up with your own sequence and calculate how many hydrophobic contacts there are to see if that sequence is a preferred protein-like sequence or not. Now that we know how to calculate the hydrophobic contacts, we obviously want to find the structure that's going to minimize the hydrophobic interaction energy. And the lowest energy state is now going to be defined as the native state of your protein. We can also find the probability of finding the chain in its native structure using this expression. So the probability of a folded protein is the statistical weight of its native state over the total weight or multiplicity. Looking back at our bottom sequence, let's try to calculate the probability of finding that U or pie shape form. We have the weight of the U shape over the total weights of all the shapes. So the weight of the U shape is simply here, e to the power of negative beta 2 epsilon, and the total weight is all three of these weights added together. This beta over here is actually called the thermodynamic beta, and that just means beta is equal to 1 over kBt. So we use that quite a bit for partition functions, but just keep in mind your thermodynamic beta is going to be a term related to a thermodynamic system. So we can now calculate the probability, and that means we can graph it. So if we plot probability versus temperature to see the chances of finding a folded protein at a higher temperature or a lower temperature, we find out that the higher the temperature gets, or the higher your thermodynamic beta, the chances of getting a folded protein at a higher temperature is very slim. And it makes sense because the higher the temperature you get, the more likely that your protein is going to be denatured and fall back into a coil state. So now, how do we approach a problem that asks us to find all the protein-like sequences? Keep in mind this is totally different from what we did before. Before, we were given a sequence and we had to determine whether it was protein-like or not. What if we're just given, for example, 6 monomers, and we have to design protein-like sequences? So keep in mind there could be more than one answer, but the common thing to look out for is that there has to be a unique native structure, and that there should be at least one preferred hydrophobic contact, because it means that at least one of the contacts is favorable and you're trying to create a hydrophobic core at the center of your sequence. So we can create a general approach to these problems now. Why don't we choose 2 residues that are in contact, because we know that that has to be favorable in our chosen U, S, or G structures, and then we can choose H or P for all the other residues so that no favorable contacts are made. So let's try this approach with a few examples. Remember, we were presented with a 6-mer toy model, so we're going to use that to help figure out how many sequences are protein-like. So these are the answers, and I'm presenting them right away because we want to try and get to them. So in the G shape, there are 3 protein-like sequences, and those sequences are defined here. For S, we have 6, and for U or pi, we have 9. So when we take a look at this, it seems like the pie shape here is going to be the best compact structure we can use to model proteins, because there are 9 possible protein-like sequences that can be created for it. So when we go forward to computational models, we would most likely try and choose a model from here because it's the most probable and the most likely, and the one with the most number of protein-like sequences. So let's start together with some general steps. The question is, how many protein-like sequences are there for a 6-mer? if we really had to analyze all the possibilities, you'd have to draw 64 possibilities, which is way too time consuming. So we have to introduce a shortcut or a better way to do so. So let's first determine how many compact structures are possible. For a 6-mer, you have your PHYS and G shapes. For an 8-mer or for a 10-mer, they might be different. In the next step, we're going to pick one of the compact structures. So we're going to pick the S shape. We're going to assign one favorable contact. So remember, you always want to have at least one favorable contact. So we force, let's say 3 and 6 to be our favorable contacts. And the rest we can just keep them as hydrophilic. So we've already assigned our H pair for 3 and 6. How many unfavorable contacts can come from this? So I'm going to draw my unfavorable contacts in blue. 1, 2, 3. So there's 3 unfavorable hydrophobic contacts. If I change this to another H, you get an additional 3 unwanted contacts. If I change this to H, I get 3 plus 2 5 unfavorable contacts. So you can try different combinations as well, but you kind of get the idea that if I keep changing or adding more and more hydrophobic molecules, then I get more energy penalties for every exposure to a hydrophilic entity. So what did this tell us? This tells us that there's a lot of possibilities and we have to narrow them down. So it would be really annoying for us to draw all 64 possibilities. We've already narrowed it down so that we have to force at least one hydrophobic contact within the molecule. Now we have to look elsewhere. We know that we have 3 separate shapes. We can actually remove the possibilities where favorable contacts are possible in other compact structures. So to reduce our workload, we take a look at the U shape and we know that there are possible interactions between 1,6 and 2,5, and these are preferred contacts. For the G shape, there are possible preferred structures with 1,4 and 1,6. So if we assume that these favorable contacts are going to be analyzed in the U and the G structures, then we can forget about them for the S structure. So that means we just take a look at the 3,6 preferred hydrophobic contact. The 1,4 we can ignore because it's going to be covered over here. So that way we can narrow down our selection and draw less diagrams and less sequences for each of our shapes. So no matter what method you choose, you usually end up with a table of options. So like I said before, we were only going to force a preferred contact from 3 to 6. After that, we have 6 different possibilities of hydrophobic contacts here. And that means we have 6 possible sequences which matches the solution we were given at the beginning. So let's take a look. We have 6 protein-like sequences because of the 2 favorable contacts. And we'll explain this much later through the live lecture where we go through an example to try and calculate why this is 6 protein-like sequences and how we can consider them to be unique ground states. So the last step is to double check other structures, which is a lot of work. We only looked at S, and there's still the U and the G shapes. Try and work on them as practice and come up with the same number of protein-like sequences for each. The general steps are pick one compact structure, like we did with S. Write out your sequence possibilities for your preferred hydrophobic contact. For each possible sequence, compare your energy penalties to the same sequence in other compact structures. if your chosen compact structure has the lowest energy penalty, then this sequence has a unique native state and is therefore deemed protein-like. So keep in mind, these are the 6 that have unique ground states. And therefore, you end up with 6 over here because you do have to make a comparison with all of the options for your S shape compared to all of your options for your other shapes. For any given sequence, we now define the degeneracy Gn or multiplicity Wn of the native state as the number of lowest energy or native conformations. A good protein will only have one native state because it means that their shape matters. So for a given number of monomers, n, we can try to figure out how many sequences are protein-like, which is usually 2 to 3%. Protein-like sequences will then have a degeneracy of 1. So those proteins that have a huge number of protein-like sequences are called highly designable proteins because it gives us many options to design different sequences with similar properties to the actual proteins we're trying to create. Using our knowledge of HP sequence design, we can choose specific conformations of proteins. So hydrophobic residues are usually sequestered in the interior of a bunch of residues, with polar ones on the surface facing the solvent. So with the examples we did before, did they predict the same? Looking back, all of the protein sequences with a degeneracy of 1 usually involved at least one hydrophobic contact, which forces the chain to fold in on itself and force all the nonpolar amino acid side chains to the center. So it does make sense, and our models did predict the same. So our examples were good predictions of what would actually occur in real life. Now, remember those hydrophobic contacts? They were assigned some sort of energy penalty, so we should probably define that and how it relates to entropy or energy. So with our previous discussions, we know that there's definitely a temperature dependency of energy because it'll allow us to determine whether a protein will be denatured or folded. But what about free energy? We know that Helmholtz free energy, F, is equal to U minus Ts. We know that at low temperatures, so when this term goes to a really small number, U is going to be favored for your free energy. At high temperatures, entropy is going to be favored. So we're going to define U, or the internal energy, as our energy penalty times the number of favorable contacts. And this energy penalty is negative because the negative energy penalty is going to be the opposite of that. It's going to be a preferred energy. So what this all tells us is that at lower temperatures, there should be a higher number of favorable contacts, so that the number of favorable contacts times the energy, the favorable energy, will make up the internal energy of the system. Now, looking at the magnitude of free energy costs, whether favorable or unfavorable, that value decreases as you increase with temperature, which makes sense. The higher you get with temperature, the more your protein is going to be denatured, and therefore there's going to be a huge change in your entropy. When we take a look at the free energy of folding, there's something unique as well. At hot temperatures and at cold temperatures, it's possible for denaturation to occur. So keep in mind that at some threshold magnitude of energy, denaturation will occur when it gets too cold or too hot. Now let's switch our focus and take a look at two competing views of protein folding. First, let's get our terms straight. There's going to be a term called chain collapse that I'll present in a bit. This doesn't mean chain collapse as in folding, it's actually referring to the collapse of the structure of the entire polymer chain into its native state. It doesn't have to do with denaturation. So a lot of students get confused with that, but just keep in mind, chain collapse refers to the entire chain collapsing into a condensed compact structure. So there are two models. There's the framework model, where we assume the formation of the secondary structure happens before the chain collapse. And that means folding has to be driven by local interactions because the folding happens before the whole chain collapses in on itself. There's the collapse model, where the chain collapses and then promotes the formation of a secondary structure. So chain collapse occurring first means that there's going to be hydrophobic or non-local interactions, forcing the chain to come closer together and then encouraging local interactions to occur before it folds into its final phase or final structure. So let's take a look at some diagrams to help explain this a little bit further. With the framework model, you have an unfolded protein. It folds in on its secondary structure first, and then it creates its native state. With the collapse model, you start with your unfolded protein. Due to hydrophobic or non-local interactions, it forces some sort of folding to occur, and it's that non-local interaction that causes its secondary structure, and further local interactions will cause the final folding of your protein into its native state. If we take a closer look at the collapse model, we note that in the collapse protein, the density of water is lower, and that the hydrogen bonding is stronger, meaning a stronger tendency to form alpha helices or beta sheets. If the structure is compact, it means that there is a lot less energy penalty for forming secondary structures because of local interactions. And as the protein continues to collapse and fold, there's going to be more hydrophobic gaps or areas within the protein. And because of that, it reduces competition for hydrogen bonding and actually makes it stronger than it were if it were around water. So we're now going to wrap up with a discussion about energy landscapes. And we're going to start with a discussion of something called the Leventhal paradox. So we've learned that protein folding occurs in many ways and can have many configurations. In fact, for any polypeptide chain, there are about 10 to 50 possible conformations. So if you do the math, and you can run it through some Python code or something, but in order for a molecule to try out every single conformation every 10 to the negative 13 seconds, it would take about 10 to the 30 years to see which native state is preferred. So this is the Leventhal paradox, the fact that the protein can find its preferred shape very quickly, but it actually would have taken an impossible amount of time for the protein to do it. So it's obvious we can't assume that the folding pathways are random. There has to be something to help them find the right preferred shape. And that's why we have to look at energy landscapes. The Leventhal paradox we talked about before assumes an equally random possibility that each protein structure can occur, which we all know is a very bad model and definitely cannot happen. We call such energy landscapes golf course landscapes. So you can test out a bunch of different areas with no such luck, but then you just hit that one jackpot by dropping into the hole to get your preferred structure. So that's highly chancy and just not the greatest model that we could use. The best one we can use is something called a smooth funnel landscape. So instead of having a flat landscape with just one perfect low energy system, you have a funnel system that goes like this. And that funnel landscape is going to be much easier because it takes into account the different energies of different conformations. So let's take a look. Here we have that golf course energy landscape, which we know doesn't work. If we have a funnel, kind of like these two, if we have a funnel-like folding landscape, we can say that it's under thermodynamic control as the energies of protein folding are taken into account, and therefore entropy is your main driving force. This means that any conformation can proceed through a series of downhill energy steps to its native state with no energy barriers. So as the protein is folding and folding, it's going to want to fold in a way that it tries to approach the bottom of this funnel so it can achieve its lowest energy state. Meanwhile, you also have proteins that have rugged folding landscapes, and these are proteins that are said to be under kinetic control. So there's a lot more variety, and it's harder for the protein to fall down a hill of possibilities. So just to summarize what we learned today, we looked at coil helix transitions. We used the LR model to represent the coil helix transition with C or H states. We were able to determine the fractional helicity using the weights of these states, and we looked at different ways of calculating their configurations using matrix formalism and their probabilities using their statistical weights and the partition function. We also took a look at protein-like sequence design. This one was highly visual, so we determined which sequences were protein-like and how to find such sequences from scratch, and it had everything to do with how many energy penalties you could assign to each of your sequences. The more energy penalties, the worse your structure. Last, we looked at protein folding energetics. So we had presented a competing view of protein folding. So there was the framework model and the collapse model. We took a look at the Leventhal paradox, which it would be good to know how to describe this in case it pops up somewhere else. And of course, you have your energy landscapes. So knowing which energy landscapes are driven by what kinds of forces would be good to know as well. Next, we're going to move on to nucleic acids and DNA. So this pretty much wraps up all of the protein-related talks that we're going to have. We are going to jump into enzyme kinetics shortly, and then move on to DNA afterwards. But in terms of protein folding, this would be pretty much it, and covers all of the content that you would have to know for protein-like sequence design and different models for proteins.